Hello dear students, welcome to all of you. So in our previous lecture we have seen basic aspects regarding fermentation, and in that we have covered basic things that what is fermentation and how it is carried out, and we have also covered the milk fermentation and its different phases and different cycles and different types. So in this particular lecture we are going to see abnormal milk fermentations. Besides normal fermentations, there are some abnormalities that are related to normal fermentation, and these are studied under abnormal milk fermentation. So we know that milk and milk products through prepared using lactic acid bacteria can be spoiled by growth of wild strains that produces unwanted gases or flavors or appearance defects. Okay, so the milk and milk products besides uh, produced by normal fermentation, there are sometimes that other strains of bacteria that are known as wild strains, they also produces unwanted gas or flavors or appearance defects. So basically these conditions are produced by non-spore forming bacteria responsible for fermentative spoilage or dairy products are mostly either lactic acid producing coliforms and genera of lactic acid bacteria involved in spoilage of milk, and fermented products include lactococcus, lactobacillus, leuconostoc, enterococcus, then pedicococcus and streptococcus. So besides normal fermentation of milk, there are sometimes the other group of microorganisms, they causes abnormal fermentation or spoilage of milk and milk related products by producing unwanted gas or flavors and different appearance, and this group of microorganisms, they includes non-spore forming bacteria and these are responsible for spoilage of milk as well as dairy product, and they includes lactococcus, lactobacillus, then leuconostoc, then enterococcus, then pedicococcus and streptococcus. So in addition to these microorganisms, there are a number of non lactic group of microorganisms that can produce enzymes which attack into major milk constituent like fat, proteins and carbohydrates, causing abnormal textural defect or odor or color defects. So there are other group of microorganisms, they produces some enzymes that produces these enzymes mainly act on fat, protein and carbohydrate portion of milk, and this action produces defects in odor like false smell of flavor and some color defects. Okay, so we'll see what type of abnormal fermentations these are. So in that first we are going to see gas production. So the production of CO2 by certain microorganisms in dairy product is responsible for gasiness. So production of gas, especially CO2, this property of fermentation process is known as gasiness, and this is abnormal condition as far as fermentation is considered. So gasiness is associated with acid production causing frothiness due to the associative action of acid producing bacteria with yeast, and production of gas is canned dairy product causes bulging of cans that is called as blowing, and it is highly undesirable. So you know that in most of the time the fermented products, they are used for longer time and they are packed into some tin like containers. So if this gas production activity get occurred in these packed products, like they are those who are packed in containers, so that gas will produce and that container will expand, and this is causes spoilage. So this is abnormal condition as far as fermentation and the fermentation products are concerned and their packing are concerned. So this production of gas is in canned dairy product causes bulging of cans, and it is called as blowing, and it is highly undesirable. So gassy cream is rejected for butter making. However, in Swiss cheese, a controlled production of gas gives desirable texture by eye formation. So gas is again important in some cases of fermentation, but in excess cases it is harmful, and it causes undesirable effects. Okay. So in cheddar cheese, gas production due to coliform is undesirable as it is accompanied with off flavors and that may be carried out to final product. So in some cases gas favorable gas production in small quantities acceptable, but in other cases gas production is not desirable and it causes off flavors as well as some other changes in the texture of that product. So in cheddar cheese, the gas production due to coliform is undesirable and it is accompanied with off flavors and that may be carried out to final product. So in canned dairy product, the production of gas may lead to early gas production or late blowing of cans, and the early blowing condition is due to coliforms and where late blowing is due to Clostridium species. So both these species, they are responsible for production of gas and that bulging of cans in canned foods, and these species are coliforms as well as Clostridium species, and the mainly causative organism for this gas production abnormality in fermentation, these includes coliform group, and that coliform group includes E. coli, then Escherichia aerogens, which ferments lactose of milk or cream into gas and acid. So these are called early gas producers that lead to blowing conditions. Then anaerobic spore forming bacteria, example Clostridium butyricum, Clostridium sporogenes produces only gas under anaerobic conditions and mostly prevailing in canned dairy products like processed cheese, concentrated milk, and these are called late gas producers and produces late blowing conditions. So lactose fermenting yeast produces CO2 and small amount of ethyl alcohol in milk, cream and whey at lower or below 37 degrees Celsius, for example Candida pseudotropicals. So again, besides bacteria, there are some yeast species, they also produces this gas as well as ethanol, and they causes spoilage of this particular milk and milk product. Then precaution and control. So how to avoid this gas production in this milk and milk product, or how to avoid this faulty fermentations or spoilage? So simply by avoiding contamination of causative microorganisms. So all these practices of fermentation, if you carried out in proper maintained conditions, in that you have to avoid spoilage causing entry of spoilage causing microorganisms. Okay. Then holding of milk and cream at ambient temperatures should be avoided. Okay. Then adequate heat treatment of milk should be done. Okay. So basically pasteurization process we carried out in any kind of milk product or its processing. So it is very important as far as milking and milk product is concerned. The next abnormality or abnormal fermentation type is ropiness or sliminess. So ropy formation is brought about by the growth of bacteria leading to change in its consistency of product that forms threads or viscous masses when poured. So rope basically literally it means thread like or rope like structure. So ropy fermentation is identified by formation of thread like and rope like structure, and it is sometimes viscous in its texture when poured. So ropiness is basically again abnormal fermentation type, and ropiness develops only on storage of milk and it is drawn out as fine threads and may appear a gel like consistency. So sometimes the change is so prominent that milk can be drawn into long threads. Okay. So ropiness has particularly been observed in milk held at low temperature as many causative microbes show active growth in milk at low temperature. So if you hold the milk for low temperature without its pasteurization activity, so we'll find the milk in milk the thread like structure get developed, and this thread like structure and gel like structure are nothing but ropiness of milk, and it is again carried out by some types of microbes. So at higher temperatures these ropy bacteria are overgrown by lactic acid bacteria. However, ropiness causing bacteria are not harmful to consumers. So ropiness is developed before detectable acid development, and it decreases the acidity increases. So as the ropinase is a property which depends upon acid formation in this milk environment. So as the acidity of milk get increases, the ropinase property get decreases. So which are the causative organism for this ropinase property or ropinase abnormal fermentation? So these includes gram-negative rods, in that particularly alkaligens viscouses. Then coli aerogens group, it consists of ropis strains belonging to Enterobacteriaceae family. Then Citrobacter and related genera, it includes Enterobacter aerogens, Enterobacter cloaceae, Citrobacter frondi, then Serrata merseni. Then aerobic spore formers have also been infrequently involved in ropinase. These includes Bacillus cereus, Bacillus subtilis, Bacillus circulans, and there are some lactic acid bacteria, they also causes ropinase in milk, and this includes Lactococcus lactis species, Lactis biovora arendicus, then Lactobacillus cz and Lactobacillus bulgaris. So overall these are the group of microbes, they causes ropinase in milk, and this ropinase of milk is again considered as an abnormal fermentation type. Then sources. So which sources from these bacteria, that is ropinase causing bacteria, they can enter into the milk. So because bacteria causing ropinase are generally not present in aseptically drawn milk, but it came from water, dairy equipments, dust, laden air, then coats of cow feed, and etc. So these are the basically sources of bacteria that causes ropinase, and these ropinase causing bacteria, they enter through these kind of sources, just like water, then dairy equipments, then dust laden air, dust present in air, then a coat of cows and some feed that are used for feeding that cow. So these are all sources from where these ropinase causing bacteria, they get entered into milk. So what is mechanism of ropinase? So materials that are capable of producing ropinase, these are the gum or gum like substances like polysaccharides. These gums are derived from lactose that is fermented by coliforms, and Lactococcus calice species, then Lactis biovora alendicus, and Streptococci, and Leuconostoc, and Lactobacillus. So basically the ropinase is the property or the tail-like structure present in milk. These are due to gum-like structures that are polysaccharides present. Polysaccharides, they are produced by fermentation of lactose sugar, and these are carried out by different types of groups of bacteria. And then means nitrogenous mucus-like substances. So mucins are a combination of proteins with carbohydrate radical may be produced by peptonizing bacteria, that is micrococci, gram-negative bacteria, and aerobic spore formers. Then there are some exopolysaccharides, they are also produced as a capsule associated with cell or as a slime layer, and these are mostly produced by alkaligens viscous bacteria. And so all these factors, they are considered as an important factor for formation of ropinase in milk or abnormal fermentation. So significance of ropinase. So ropinase causing bacteria are not harmful to consumers. Okay. So it's good that these bacteria or ropinase property is not harmful for consumers. However, this defect affects acceptability of a product that occurs less frequently in pasteurized milk. But the ropinase milk is rejected, it not get accepted as a part of quality control. So ropinase has particularly been observed in milk held at low temperature. At higher temperature, lactic acid bacteria dominate ropibacteria. So ropinase is observed higher in summer, more prevalent in spring and autumn. Okay. So prevention and control. So how to prevent this property of ropinase? So clean milk production and effective heat treatment removes all these ropinase causing bacteria. Then post pasteurization of contaminated should be avoided. Okay. So these are the two factors due to which we can control the prevention and entry of these bacteria. Then next is sweet curdling. So curdling without pronounced acid production is sweet curdling. Okay. So this defect is due to the production of an extracellular enzyme similar to rennin by bacteria that precipitate casein before development of sufficient acidity. Okay, so curdling is what? Curdling is acid production. So curdling is curdling without pronounced acid production in Swiss curdling. Okay, so this defect is due to production of an extracellular enzymes. Okay, similar to rennin by bacteria and that precipitate casein before development of sufficient acidity. So this defect is commonly observed in milk and cream, and particularly that are moderately heat treated, particularly when held at higher temperatures during summer month. So rennin like is produced generally by aerobic spore formers. So the activity of this enzyme is known to increase at higher temperature. So little or no acid production is observed during sweet curdling. So under Indian conditions, it is observed that boiled milk, if allowed to cool and stand in same container for a long time, that will produces undergo sweet curdling. So this defect sometimes becomes apparent on reheating of the product. So the defect may also occur in pasteurized milk if stored for a longer time under refrigerated conditions. So under farm conditions, milk contain microbes, but the defects does not appear as commonly encountered lactic acid bacteria curd the product by acid production. So what type of microorganisms they cause is curdling of this milk. So these includes cocci, especially streptococci liquefaciens. Then there are some aerobic spore forming bacteria. These includes Bacillus cereus and Bacillus subtilis. Then psychrophilic spore formers. These includes Bacillus cereus, Bacillus licheniformis, and certain microbacterium species. And there are some noospore forming rods also. This causes curdling, and this includes Proteus species as well as E. coli. So significance. So what is the significance of sweet curdling? So sweet curdling is prevalent in heat treated products, particularly in summer. However, it is observed in refrigerated and boiled milk stored for longer duration. So factors affecting sweet curdling. So high temperature is more prevalent in summer. So age of milk. So these are the factors, just like high temperature, age of milk, and pasteurization. So these are factors that affect sweet curdling process. And then precautionary and controlling measures. The best way to avoid curdling is to avoid contamination. Then control storage temperature and pasteurization at temperature higher than normal. The next abnormality is proteolysis. So proteolysis is the process where proteins get breakdown. Okay, so it is the process in which casein or some insoluble casein derivatives are broken down to water soluble compounds through the action of microorganisms or their enzymes. So as you all know that in milk casein is the principal protein, and in milk the casein and some other proteins which are present in insoluble form, so they all are degraded by the action of microbes and their enzymes. So milk contains different proteolytic enzymes that may bring about alteration of milk constituent. Some of these are naturally present in milk, while others are elaborated by microorganisms. So since milk contains only small amount of non-protein nitrogen substance to sustain prolonged growth or survival of microorganism depends upon enzymes that hydrolyzes milk protein. So these hydrolyzing enzymes, they are already present in milk, or sometimes they are produced by microbes. So changes in milk proteins as a result of cyclotroph growth or enzymatic action are important in keeping quality of milk and milk product. Okay, so release of various nitrogenous compounds or degradation of individual protein fraction is observed while studying proteolysis caused by cyclotropic enzyme. So basically this proteolysis activity or breakdown of protein molecules is caused by cyclotropic group of bacteria and their bacterial enzymes. So raw milk supplies contain heat stable proteases or microbes that are able to produce. These proteases can attack casein and where proteins, leading to coagulation of milk and development of bitter taste. So these are the side effects of protein breakdown. And proteolytic flavors are attributed to breakdown of proteins and amino acids, and that proteolysis is often accompanied by development of bitterness due to presence of peptides that are bitter in taste. So basic effect of proteolysis is bitterness in milk. So proteolysis causes bitterness of milk. Okay. So cyclotropic proteinases primarily act during refrigerated storage of raw milk before and heat treatment. So ultra high treatment sterilized milk coagulated in presence of heat resistant Pseudomonas proteases. Okay. Then causative microorganisms. So cyclotrophs are the active proteolytic that grow at 7 degrees Celsius or less. Especially this cyclotrophs group includes Pseudomonas fluorescens, Pseudomonas fragi, then Alteromonas putrefaciens, then Thermodurix. These are especially Micrococcus cazuliticus, then Bacillus stereothermophilus, and Bacillus cereus, and Bacillus subtilis. So all these are microorganisms that brings about breakdown of protein, and that process is known as proteolysis. Then lipolysis. So just like proteins, there are lipids are also present in milk, or fats are also present in milk, and these fats they get broken down by the action of some enzymes present in milk, and some enzymes that are secreted by microbes. So it is the hydrolysis of milk fat and subsequent production of off flavors in dairy products, and this process is known as lipolysis. So lipolysis is carried out by lipase, resulting into accumulation of free fatty acids. So lipolysis, that is breakdown of fat, and this is caused by, or this process of breakdown of fat or lipids, it's carried out by an enzyme that is called as an lipase, and this lipase acts on fats and lipids, and it causes breakdown of fats and lipids, and after breaking down of fats and lipids, the free fatty acids they get released, and this causes accumulation of free fatties, and this causes off flavors. So the lower chain fatty acids, particularly butyric and caproic acids, are responsible for lipolytic off flavors, and the onset of lipolysis in milk at any stage from milking, storing and processing should be avoided, since rancid milk or cream will not yield product of flavor quality. So the intrinsic milk lipase is present in sufficient quantities to cause hydrolysis of milk fat. However, fat globules membrane protects the milk triglyceride from attack by lipolytic enzyme. So if you see milk composition, so the lipase enzyme is already present in milk, but it is present in very smaller quantity, and the fats they are covered by fat globules, and these fat globules prevent lipase from being act on that fat material. Okay, so whose activity is further impaired by association with casein, and possibly by the presence of lipolytic inhibitors. Okay, so the intrinsic milk lipase is well partitioned from triglyceride substrate, is little or no lipolysis occurs in milk, but lipolytic microorganisms and their enzymes produced in bulk cooled milk, cream by cyclotrophs, and survivors in HT-ST pasteurization and UT sterilization causes lipolysis during subsequent storage of heat processed milk even at low temperature. So there are some group of microorganisms that causes lipase enzyme, and they are still get survived after different stages of pasteurization, and these are the actual group of microbes that produces lipase enzyme, and that lipase enzyme is responsible for breakdown of fat as well as other fat related molecules. So however, in some instances lipolysis appears to have favorable influences on flavor of cheese. So this lipolysis process is, if you see in some products, so it is favorable, but in some products it causes off flavors. So lipolysis is not desirable to our products. Okay, so hydrolysis of cheese constituent is essential for development of mold ripened cheese. So not only the lipolytic activity of mold, but also the further metabolism of fatty acids, especially their oxidation to methyl ketones. So in addition of lipase of Candida lipolytica improves the quality of blue vein cheese made from raw or pasteurized milk. So what are the causes of lipolysis? So intrinsic milk lipase. So these are the enzymes that are intrinsically means already present in milk, and these lipase enzymes, they break down the lipid or fat molecules. Then lipolytic enzymes of microbes. So these are the group of microbes. Here you can see different types of cyclotops, then other types, and yeast and molds. So all these microbial groups, they secrete enzyme lipase, and this enzyme lipase brings about breakdown of fat as well as lipids. Then how to prevent this lipolytic activity? So simply by clean milk production practices, or cooling of milk as per recommendation, or processing of milk prior to microbial growth. So these are the some controlling and preventive measures that can use for avoiding the lipolytic activity. Then next is Briti cream defect. So Briti cream or broken cream is a well known example of effect of spore formers of milk. So this defect which occurs in pasteurized and raw cream is characterized by appearance of flakes in cream layer that do not mix even when milk is shaken, and in such a milk is used in tea, and the flakes float on surface, making is unacceptable to consumers. So BT cream defect, so it is nothing but formation of flakes of fats or some fatty layer on milk. So these flakes are of two types, one is mechanical and other is physical origin, and there are flakes occur when flat globules membrane is partially disturbed and stick together. So this condition is the second type. These changes in formation of flakes, these are brought about by mechanical or physical way, or there are some microbes that causes some enzymes that are known as lecithinase enzyme, and these are produced by Bacillus cereus, Bacillus and other group of Bacillus, and so this property is caused by microbes as well as physical handling. So again these are precautions like clean milk production and avoid spore contamination. So these are the best way to prevent this property. Then fermentation based on discoloration. So the number of different abnormal colors are developed depending upon growth of microorganisms, and they are listed below. Yellow fermentation, it is caused by Pseudomonas cyanantha. Then blue color, then it is caused by Pseudomonas cyanogens. Then black color, it is caused by Pseudomonas nigrificans. Then red color, it is caused by Serita marcescens. Then green color, it is caused by Pseudomonas fluorescens. So these are colored products that are usually seen in milk, and these colored product or discoloration, so these are considered to be the abnormal fermentations or spoilage of milk, and all these are different colors of milk. They are caused by this type of different microorganisms. Then abnormal flavors. So most of the times we often get off flavors or abnormal flavors from milk and milk product, and these are considered to be the faulty or defected or spoiled fermentations, and these are known as fruity flavor, malty flavor, then bitty flavor, then fishy flavor and potato flavor. So all again these are different types of flavors, and these are caused by different types of microbial groups. So abnormal flavors, so most of us off flavors are microbiologically originated. So basically the off flavoring property in milk and milk related products, so it is basically related to microbes. So these off flavors, they are carried out by microbial changes. Okay, so microbial off flavors are invariably encouraged in raw milk through occasional neglect or failure in refrigeration. So basically these kind of off flavors we can see in raw milk. Okay, so improvement in processing operations has resulted in a lower bacterial count, and hence most microbial spoilage and associated off flavors can be tracked in post pasteurization contamination with cyclotrophs. So these are the types of flavors, and these are the respective microorganisms that causes these flavors. In that you can see fruity flavor, it is due to ethyl ester formation, and it is usually catalyzed by esterase produced by saccharotrophs or lactic acid bacteria, then ester formation by Pseudomonas fragi, then multiflavors, it is caused by multistrains of Lactococcus lactis species, then Briti flavors, it is caused by proteolytic microorganism, especially bacillus species and Pseudomonas species. Then fishy flavor, it is caused by Pseudomonas species. Then potato flavor, then phenolic flavor, then musty flavor, then burnt of clamal flavor. So these are of flavors or different flavors, and these are caused by different types of microorganisms. So here we have finished some spoilage or abnormal fermentations. Okay, thank you.